10 o'clock. So Carolyn, you ready?
I'm ready.
All right, so we're going to go ahead and get started. We've got Dr. Carolyn Hodo with us today. We've got a couple folks coming in. Come on in and have a seat. Dr. Hodo has a bachelor's from Berry College, a DBM from the University of Georgia at Athens. She did her residency at A&M in the Texas Vet Diagnostic Lab. And she'll tell us a little bit about that, I would imagine. And then a PhD here as well. She's currently a postdoctoral fellow at A&M as well. So she's going to give a seminar on some of the work she's doing here and her experience. So welcome.
Thank you. Thanks so much, everyone, for coming. Yeah, I'm going to start a little bit about my background. So as you mentioned, I went to Berry College, which is a tiny college in northwest Georgia, where the deer actually outnumber the students 8-1. So there's about 2,000 students, and there were 8,000 deer. They have a huge campus, mostly wildlife management area. I did a little bit of research there, studying how deer respond to sound, and also looking at hemoparasites in bluebirds. From there, I went to University of Georgia for vet school. During my 4th year there, I did a couple of interesting research experiences. I did 2 externships at the Southeastern Cooperative Wildlife Disease Study, or SCWDS, where I got to experience mist netting, which was super fun, and then learn about other wildlife diseases and do wildlife necropsies there with the pathologist. I also traveled to Costa Rica for a few weeks to work on a project looking at zoonotic diseases in domestic dogs in indigenous villages, and then catching some local wildlife around the research center there. Came to A&M to do a pathology residency, which, as Roel mentioned, was funded by the Veterinary Diagnostic Lab, but I was housed mostly in the vet school's necropsy and visited diagnostic lab to do necropsy service a few weeks. I did a few wildlife-focused case reports that I won't talk about today. One was really interesting. New for Texas species of Sarcocystis in doves, in wild doves. And then some interesting fungus in sea turtles. These are actually captive sea turtles at the NOAA station in Galveston. So during my residency and then afterwards, I started working with Dr. Sarah Hamer in the vet school to do a PhD. Started off with helping out with a project on sandhill cranes, looking at sandhill cranes as a surrogate for the endangered whooping crane, to see what parasites they might have. So we did quite a few, traveled around the state to collect hunter-harvested sandhill cranes and do necropsies on them. Did some more mist netting. And this is me demonstrating bird processing techniques to epidemiology students. Did a little bit of work in laboratory mice. Several dog projects looking at Chagas disease in domestic dogs. This one on the bottom right was actually just 2 days ago down near Victoria, visiting a kennel to test dogs for Chagas and look at, that's a portable, like phone-based EKG monitor. So we can read the dog's heart rhythm. And then some other wildlife work, besides what I'll talk about today, in the Sam Houston National Forest, looking at ticks and tick-borne diseases in mammals there. So brief outline. I'll talk about my existing research characterizing reservoirs of vector-borne diseases, specifically within the Trypanosoma cruzi system. And then one other interesting research project that's sort of breaking news. Then a little bit about my vision for research at AgriLife Uvalde, and some specific research ideas. So I studied disease ecology or epidemiology or ecological epidemiology. It's all sort of just the study of interactions between wildlife, between hosts and their pathogens and the environment that they're in. So this is the epidemiology triangle with the vector in the middle. And controlling these diseases with complex transmission systems requires knowledge of all of the aspects of this triangle and the interactions. And then those interactions can be targeted to break transmission. So in vector-borne diseases, you can target the vectors in the environment with like larvicide for mosquitoes, for example. You can vaccinate the host to prevent the host from, the agent from acting in the host, for example. So part of these complex systems is identifying what is the actual reservoir of disease. And so the reservoir is the population or species in which the pathogen is maintained and from which it's transmitted to the target population. So For example, like West Nile, there's a bird, usually bird-mosquito cycle, and then there's spillover to dead-end hosts, humans, horses, sometimes dogs, but sometimes the target population. So the target population is just the population that we care about of interest. So sometimes it's humans, sometimes it's endangered wildlife species, sometimes it's domestic animals. So just in this context, that's the one. Sometimes the target population actually acts as a reservoir itself. So it's important to note that just the ability to be infected by a pathogen doesn't necessarily make a species a reservoir. So you can identify a pathogen in an animal, but that doesn't mean that that animal is actually infectious to infect a vector or infect another species, another individual. And in vector-borne diseases, animals can be infectious, but if they don't come into contact with the vector, the same vector that's potentially going to transmit to humans, they might not be important as a reservoir. Also, some simpler definitions of reservoir say that the reservoir is the one that's not affected by the disease, but that's not always the case. They just have to stay alive long enough to transmit the disease on. So how do we quantify reservoir importance? As part of a review paper, looking at this in the context of Trypanosoma cruzi, I sort of redefined this mathematical equation that if you had all the data, you could plug in values for this and calculate reservoir potential. So looking at, first on one side, the host vector contact index. So looking at feeding preference, if there's a given, if the vector had a choice of any available host, if it preferentially feeds on one or another, and then multiplying that by the available relative abundance of hosts in a given environment, and then the number of vectors successfully fed by an individual host, which takes into account host behavior, such as actually do they eat the vector, do they have avoidance behaviors that make them less likely to be successfully fed upon by a host. And then multiply that by the reservoir competence, which looks at the mammalian, or usually reservoir. So the probability that that host is infected, so the prevalence in that population, and then of those that are infected by the pathogen, how many are actually infectious? So how many are capable of infecting another vector or another animal? And then there's a transmission efficiency from host to vector. So even if a vector feeds on a host that's in theory infectious, not every single vector that feeds on it will be infected. So if we had, in an ideal world, all of this information, you could calculate, okay, this reservoir is more important than that one, considering all these factors. So my research focused primarily on these two, the prevalence and then the infectiousness of those species, and a little bit on feeding preferences with blood meal analysis of vectors. So this system that I worked in, Trypanosoma cruzi, it's the causative agent of Chagas disease, which is a devastating disease affecting approximately 6 million people worldwide currently. This map shows it's mostly a problem in Latin America. It looks like it just cuts off at the United States, but that's not entirely accurate. There is local transmission in the United States. An estimated 300,000 infected people live in the US. Most of those are probably immigrants from these more endemic countries, but there is local transmission as well. It's a vector-borne disease. These are known as triatamines, also colloquially known as kissing bugs. And unlike some vector-borne diseases, which are passed in the saliva, in this case, the parasite is passed in the feces of the bug. So the actual act of blood feeding doesn't transmit the parasite, but often the bug will defecate on the host, and then the feces are rubbed into either the bite wound or into mucous membranes, and it's transmitted that way. Also, in a lot of cases, we think in wildlife and domestic dogs, it's transmitted through oral ingestion of the bug. And that can happen in humans as well, where unpasteurized juices are contaminated by the bug. There can be oral transmission. It's a generalist parasite that's been reported in over 150 different species of mammals. And disease is most commonly described in humans, dogs, and non-human primates. So it has 3 disease stages. So the acute stage, immediately after infection. Sometimes there's mild symptoms during that phase, flu-like symptoms. Sometimes there's nothing at all. Rarely, affected individuals will actually die during the acute stage of heart failure. But then most of those that are infected will pass into this indeterminate stage, which is relatively long, 10 to 20 years in humans, during which no signs are present. And a proportion of those, in humans it's about 30%, will progress to the chronic determinate stage or symptomatic stage, where they will, the disease is generally cardiac disease. It's manifested by arrhythmias, inflammation of the heart with dilation, and then ultimately heart failure. So this is a pretty severe case in a dog from the A&M necropsy archives. So normally, this is the right side of the heart, this is the left. Normally the right side is much smaller than it, whoa, go away. It would normally have a line like right here, and this is extremely dilated. And then there are also gastrointestinal syndromes that manifest most commonly in South America. It can be like megaesophagus and megacolon. There's 6 different genetic strain types, TC1 through 6, and they have different geographic distributions and potentially different disease outcomes. There's also a 7th known as TC bat that was described recently that's associated with bats. It's been isolated from one human so far, but it was also co-infected with another strain, so they don't know if it was causing disease. And you'll see in the US, we have both type 1 and type 4 primarily, or exclusively. So we have a long history of T. cruzi in the US. There's been some talk in the media about, oh, it's spreading, it's coming in, the vectors are spreading, but it's actually been here for a very long time. First reported in 1916, which is only 10 years after it was the parasite was discovered in Brazil. There have been wildlife reports since the 1940s. The first locally acquired human case reported was in Texas in Corpus Christi in 1955. And then the first published dog report was out of Texas A&M in 1972. They had a whole case series of dogs. So as I mentioned, we have strain types 1 and 4. And in the locally acquired human cases, only strain type 1 has been isolated. But it's still kind of a small sample size of humans that have been strain typed. Dogs, it's a major problem in dogs. Lots of kennels in Texas are with dogs that are housed outside have huge problems where their breeding stock is decimated by this disease. And Texas does appear to be a hotspot, if not for disease transmission, then at least for disease reporting or disease detection. It's also a big problem in non-human primates and animals that are housed outdoors, as we have several of those such facilities in Texas. There's very limited options for disease management. There aren't any vaccines. People are working on them, but right now there's nothing. There's also very poor options for treatment. So the drugs are relatively ineffective in the chronic stage and not widely available in the United States. So because it's hard to treat the disease or prevent the animal from becoming infected, it's better to break the transmission cycle to prevent infection before it happens. Using environmental management, vector control, and potentially reservoir-targeted interventions. So just a simple transmission cycle. In the southern United States, we have a robust sylvatic transmission cycle. So it's propagated in wildlife and the kissing bug vector in the sylvatic area, which sometimes butts right up to the houses. And then there's spillover to these paradomestic settings. So people's homes and dog kennels are, as I mentioned, are a pretty big issue where the dogs are outside. The bugs have a ready source of blood meal. There's also a few other, so it's been pretty well described in possums and raccoons, but there's some other wildlife species where the parasite has been detected, but their role as reservoirs is less certain. So the first of these I'm going to talk about is bats. So bats, you may know, are well-recognized reservoirs of many zoonotic diseases. I think there's currently a Nipah virus outbreak going on in India that's associated with bats. They also undergo long-distance migration, so there may be some potential for bats that migrate to other countries to move different strain types around. They've long been associated with trypanosomes. So T. cruzi is within the Schizotrypanum subgenus, and all of the other trypanosomes within that subgenus are only found in bats. And T. cruzi is the only one that's sort of broken out to infect other species. And so there's then sort of this bat seeding hypothesis is a new thing that's come out in the last few years where they think that Trypanosoma cruzi evolved as part of a bat cycle. So bats have been reported to be infected with T. cruzi in Central and South America and are proposed to be important reservoirs, but we wanted to know, are they reservoirs of T. cruzi in the United States, which hadn't been investigated or hadn't been published at least yet. So 2 components to this study. The first was what we term peridomestic bats. So these are bats that were encountered mostly by members of the public. Usually they're dead or dying. And then people send them into the State Health Services Department for rabies testing. So they receive lots of these bats every year, hundreds of bats. And so we receive carcasses, and we're able to collect the hearts and a little bit of blood sometimes from them. Well, we got a total of 474 bats from 29 different counties around Texas. Also, we wanted to target bats, free-flying bats in more sylvatic areas. So we sampled bats in collaboration with Jessica on East Foundation properties down in South Texas. We set up mist nets over cattle watering tanks. So as the bats would come in early in the evening to get a drink of water, they would hopefully get caught in our net. We got a few bats this way, but not a huge sample size. So we also reached out to a collaborator in Uvalde, John Barnes, who some of you may know, and he hooked us up with the Frio Cave, which is just north of Uvalde, and they have it's a cave that's full of Brazilian free-tailed bats. So instead of setting up a mist net in the mouth of that cave, where we would probably catch 1,000 bats all at once and not be able to deal with them, I constructed these handheld mist nets. So these are PVC framed, and there's a mist net strung between them. Okay, so it's just a short video. So then we sampled blood from the uropatagium, just punctured it from a vein in the uropatagium, punctured it with a needle, and sucked up about 20 microliters of blood with a hematocrit tube. And these bats were released alive. So in total, we got nearly 600 bats from 8 different species. The vast majority were Tetrarubia brasiliensis, the free-tailed bat, which was about 80% of our sample. And then the next most numerous were the evening bats, Nycticius humoralis. And then we got just a handful of bats from these other species. So after PCR analysis of the heart or the blood, only one of those bats was infected with Trypanosoma cruzi. And it was an evening bat from Hidalgo County that was submitted to State Health Services. But we did find 9 bats infected with Trypanosoma dionysii, which is a bat-associated trypanosome that had not previously been reported in US bats. So that was interesting. And then even more interesting was this Blastocrithidia species, which is an insect. It's so far only been associated with insects. Insect-associated trypanosome. And when we constructed a phylogenetic tree, the T. cruzi grouped with strain type 1, which, as I mentioned, is the one that may be more associated with human disease and more likely to cause cardiac disease. So that's interesting, even though it's only one. The Dionysii did group with other New World isolates, but it wasn't identical to any of the previously deposited sequences in GenBank. And then those Blastocrithidia species were pretty diverse, but some of them were similar to isolates that were detected in triatamines, so the kissing bugs, in Texas, so another research project that had happened a few years earlier. So probably, given the very low prevalence, these are not very important reservoirs of T. cruzi in Texas. It was an overall 0.2%, or just in the evening bats, it was still only 1.4%. But it's possible that because the reservoir community can be composed of multiple species, they could comprise part of that. And so in the context of the total bat population size, it was hard to get a good estimate for evening bats, but from what I got, if you extrapolate that 1.4%, maybe there are about 1,400 to 14,000 infected bats across the whole US. And there was a paper published a couple years ago, blood meal analysis, where they did show that a triatamine bug had fed on an evening bat. So that shows that there's contact between those species. And we did have those other interesting trypanosome findings, which I sent some samples down to some people in Brazil that are looking at trypanosome evolution. So, moving on, the next species we investigated were coyotes and raccoons, 2 very common wildlife species in Texas. Raccoons have been known to have a very high T. cruzi infection prevalence, up to 70%. They're almost exclusively associated with strain type TC4. And of those that have been investigated through pathology, there's very minimal disease that seems to develop. Coyotes are a little bit less well studied, only seroprevalence studies up to this one, looking at, well, except for, one other one that came out of our lab, 4 to 14% prevalence, so a little bit lower. And no one has done strain typing on these guys, and there's been very limited pathology investigation. So I wanted to compare the infection dynamics between the species, look at strain type associations, as well as look for pathology that develops. So we took advantage of this varmint tournament that happens in Lexington. So it's a 24-hour hunting event where teams of hunters come together. They all disperse at noon on the first day, come back 24 hours later with their hunted animals, which is going to be raccoons, bobcats, coyotes, and foxes. And so we were there when they came back to receive samples, as long as they agreed, to donate the carcasses, and we collected hearts and blood from those animals. So it was a great way to get a big sample size in one day. We got like 97 coyotes just in that afternoon. Also, we collaborated with Department of State Health Services and Wildlife Services to get samples from some coyotes that were culled as part of the oral rabies surveillance program down in South Texas, and I think they were in Laredo. And we got the whole heart from the coyotes and raccoons in Central Texas, and we just got the apex, so a tiny piece of the apex of the heart from those coyotes in South Texas. So we dissected them, and looked for gross pathology in the lab, collected samples from each of the 4 chambers of the heart, and then processed those for histopathology and PCR. And for the South Texas coyotes, we were only able to do PCR. We got DNA from those and did a couple of PCRs to screen for T. cruzi and look for strain type. In coyotes, the overall prevalence was 8.2%, so 10 out of those 120 coyotes. A little bit higher prevalence in South Texas, although we had a smaller sample size. And they were PCR positive in either the heart or the blood, but never both. Like the same animal didn't have both heart and blood positive, which could support that during the acute phase, the parasite's circulating in the blood, but then during the chronic stage, it may not be circulating, it's just present in the heart. In contrast, the raccoons had a higher prevalence, 63%. 52% were positive in the heart, and then 44% were positive in the blood, and almost, all those 44% were also positive in the heart. So even though they have the parasite in the heart, they're still circulating it in the blood. And they were spread across, the raccoons were spread across the counties, while the coyotes were sort of more associated with the more southern ones. So I did histopathology on all these as a pathologist. I wanted to look at, see what I can see, what inflammation is present. So I examined a section from each of those 4 chambers of the heart and gave it a numeric inflammation score. And then I re-examined those that had a higher inflammation score to actually look at, okay, is this inflammation consistent with Trypanosoma cruzi, or is it more likely to be something else, or can I identify another parasite? So there were the coyotes that were PCR positive had a much higher average inflammation score than the PCR negative coyotes, and then the raccoons. And the raccoons showed no difference, no significant difference between positive and negative. Wow. So this is a histopathology slide. Don't know how many of you are used to looking at this sea of pink. This is the heart. These are all individual heart muscle fibers. And then these darker blue areas are inflammatory cells. So these are all inflammatory cells, lymphocytes, and plasma cells. In this coyote, there's destruction of the normal heart muscle cells. So they're replaced by this pretty intense inflammation in this focal area. In contrast, this is a slide from a raccoon, and this was actually the very most severely inflamed raccoon, which is really very mild. Like normally, if we were reading this slide not specifically looking for this inflammation, we would probably just not even report it. So the inflammatory cells are there, but they're not really destroying the surrounding muscle cells. So I mentioned I did find some other lesions in these coyotes, so always looking at wildlife, they're likely to be infected with multiple parasites. I found microfilaria of the canine heartworm, both within vessels, and then there are microfilaria in here associated with a granuloma. And then another protozoan parasite, Hepatozoan americanum, forms these cysts, and then when they mature, they can release zooites that are also associated with similar inflammation. So in summary, for that project, coyotes, although they're less likely to be infected with T. cruzi than raccoons, lower prevalence, they did exhibit more severe pathology, and we found strain type 1 in the coyotes and strain type 4 in the raccoons. So the question is is the pathology dependent on the strain type or the host species? So does this support the fact that strain type 1 causes more disease, or are raccoons just adapted to not develop disease, either way? As far as the reservoir status, there was a much higher prevalence and higher parasitemia in raccoons. But as I mentioned, the coyotes harbor TC1, so raccoons are certainly good reservoirs for TC4, but coyotes might be of more importance to human health. There is likely a risk to hunters that handle these carcasses and handle the blood. If the parasite is circulating in the blood, for sure transmission could occur through, if the hunter had a break in his skin. And then the population level significance as far as the health of the wildlife is unknown. There was that inflammation in the heart of the coyotes. It's not incredibly severe, but sometimes it doesn't have to be to interrupt the heart rhythm. However, these were apparently healthy coyotes that were shot by hunters. And if there were more severely affected ones, we may not ever find them. So it's difficult to assess health status in free-ranging wildlife. All right, so third and final T. cruzi related project is related to non-human primate facilities. So as this parasite spills over from the sylvatic cycle into peridomestic, human housing, it also spills over into non-human primate facilities. So it's an increasingly recognized problem at these facilities in the southern US. The monkeys are housed outdoors. They don't have any bug screening protection. And it's a major economic concern because these are very valuable monkeys, and once they're infected with T. cruzi, they're not as useful for other research studies. And so this is a known problem in these facilities, but the transmission details were not well characterized. So we were approached to sort of look at what's the ecology at these facilities, and are the local wildlife involved, how many vectors are around, some of these facilities were concerned that the non-human primates themselves were just transmitting it directly to each other without the role of the vector. So we looked at both the vectors, or the reservoirs, and the non-human primates themselves, and whether the non-human primates are transmitting back to, or potentially transmitting back to the vectors at 3 different primate facilities across Texas. One in San Antonio at Texas Biomed, the Southwest Primate Research Center, down in Alice, Texas at SMBL, and then in Bastrop at the MD Anderson Cancer Center. And so we did slightly different amounts of work at each of the studies, and the one in Bastrop was sort of the most comprehensive project where we looked at all 3 components of the transmission cycle. So first, starting with the vectors, we did passive vector surveillance or collection where we educated the facility personnel, what do these bugs look like? What do you do if you find one? And so they were collecting them for us. And then we also visited 2 of these facilities to do active trapping of bugs. So kissing bugs are more challenging to collect than, say, mosquitoes, where you can't just set a trap and then come back and check it in the morning. So we set up UV lights and white sheets and then actively monitor them to see if any bugs came into the sheets. And we also searched around the facility with flashlights at night and looked for bugs. Then those bugs were dissected in the lab, DNA was extracted, and we did some PCRs for infection status and strain typing. We got a total of 78 bugs from 2 of the facilities. Most of those came from the Alice facility, and then we got 6 from Bastrop. And we didn't find any bugs during the few visits that we made to San Antonio, even though we know they're in the general area. Most of those were Tritoma gristaferi, which is the species that's most abundant in South Texas. Interestingly, at the Bastrop facility, these white top buildings, this is where the rhesus macaques are housed, and this is a shower building, and every single one of the bugs that they submitted to us came from that building. So it's kind of on, it had lights on at night. It's on the edge of the sylvatic habitat between that and the monkeys. So that may explain that. Half of those bugs were T. cruzi positive. And we looked at strain types, so most of them were infected with TC1, but quite a lot with TC4, and then several had mixed infections with both strain types. We did a little bit of blood meal analysis using just traditional PCR and Sanger sequencing, and we found diverse host species, including white-tailed deer, raccoons, a gecko, which is not a competent reservoir for T. cruzi, Canis species, so we can't differentiate between domestic dog or coyote, and then a potential human blood meal, which we were unable to confirm on a second test, so that's why it has a question mark. We didn't detect any non-human primate blood meals. We suspect potentially any bug that comes into the non-human primate cage to feed on a primate will be discovered and eaten immediately. Maybe. So moving on to the wildlife. At some of these facilities, particularly the one in San Antonio, roof rats are a very important pest species and are very numerous. So they had an active pest control program that was collecting these rats, and we received the carcasses of those, which we dissected and collected the heart and blood. We also did some active small and medium mammal trapping at the Bastrop facility using Sherman traps and tomahawk traps, and sometimes catching rodents in large tomahawk traps, which was challenging. And those animals were euthanized so we could collect the whole heart and the blood. And then we did PCR on both blood and heart. So we found that the mesomammals that we caught, the possum, raccoons, and skunks, were highly infected. So we didn't have a huge sample size from that one summer of trapping, but we got a total of 10 mesomammals, and 8 of them were infected with T. cruzi. And mostly it was both in the heart and the blood for those. So I think every single one had the parasite DNA at least circulating in the blood. None of the rodents were positive, which was a little bit surprising. Wood rats have definitely been highly associated with T. cruzi, and we only got one, so that might explain that. But then out of the 153 roof rats that we got, mostly from San Antonio, none of those had the parasite. So looking at the primates themselves, we got blood and heart from cynomolgus macaques down in Alice, and then we got blood from seropositive rhesus macaques in Bastrop. And those macaques were still alive in the colony, so we were able to do serial sampling up to 4 times if they were negative on the previous tests. So we tested them basically until they were positive, or until it had been 4 times. And then we did qPCRs on those. So in the primates, we were able to demonstrate positivity, at least eventually, or at one time point, in 75% of those in the blood. And then some cynomolgus macaques just were PCR positive in the heart. And there was a very equal mix of strain types. So unfortunately, we didn't identify any specific strain type that's most likely to affect the monkeys that would enable us to target a certain reservoir species. So they were pretty equally affected. And then there was intermittent positivity in those rhesus macaques. So I mentioned some of them we had to sample 4 times, some of them we sampled 4 times, and these were all seropositive monkeys. So they had antibodies to the parasite, they've been exposed. And they also had a relatively lower parasite concentration than we saw in the wildlife. So this violin plot, CT values are inversely related to parasite concentration. So the longer it takes for the PCR to amplify, the lower the concentration of the parasites. So this was like a 100-fold difference between the parasite concentration in wildlife compared to the monkeys. So in summary, it looks like medium-sized mammals are likely more important as reservoirs than the non-human primates and the rodents. And we saw no clear strain type associations in the primates, but we did find expected strain type associations in the wildlife. And the vectors were present. They are infected with T. cruzi. Alice, Texas is where we got most of ours. And it's possible that in the other facilities, as I mentioned, that the bugs are being consumed, and that's why they're not found by facility personnel as commonly. So moving on, I wanted to talk briefly about some other work that I've done, and this is sort of breaking news. As part of some rodent trapping projects with people in the wildlife department and entomology, both here at the Biodiversity Research and Teaching Collections in College Station and at the Atwater Prairie Chicken Refuge a little bit farther south, rodents were captured with these lumps on their tail or on their feet, and they were sort of recorded as tumor tail mice in the field notes. And it was mostly Biomys taylori, but there were a few Sigmonod hispidus involved as well. And these had these, so we collected one most severely, I think Adrian maybe got this one, most severely affected and was submitted to me for necropsy. So I was sort of looped in early. What could this be? I don't know, maybe you could get me an actual specimen. So we got it, did necropsy, got these big masses, and there were no apparent internal lesions. And so I took sections for histopathology. This is a slide. This is the mass. So this is all like skin tissue, but it's very abnormal. So it's thrown into folds. It's really proliferative. And then on higher mag, you can see these pink dots. So those are intracellular, likely viral inclusions. So we weren't sure exactly what virus. So we took advantage of the electron microscopy lab down in the basement of the vet school. And saw that this is one of those inclusion bodies, and it's filled with viral particles, which are actually poxvirus. So it has this double envelope brick-shaped inclusion. So we did a preliminary PCR and sequencing in the lab using some poxvirus primers and targeting a short region of gene, and it did not match to anything existing in GenBank. So then working with Jessica Light and with a collaborator at the CDC, poxvirus branch, we were able to do full genome sequencing and a detailed phylogenetic analysis, and found that indeed it is a novel poxvirus, likely a new genus. So this, you don't have to read all of these, but this is our virus. It's on its own branch, and it doesn't fit within any of these previously described genera of poxviruses. So we named it Brazos pox because those sites are close to the Brazos River. Population implications for the rodents. There wasn't evidence of systemic disease like some poxviruses cause, but we only had a sample size of 2 mice that we actually did a necropsy on. and was found at 2 different field sites. For sure, that one mouse that had the larger lesions was probably affected negatively, but it would be good to examine more animals. Many poxviruses are zoonotic, example cowpox, and then you may remember the monkeypox outbreak associated with prairie dogs back in 2003. And since this paper just came out last week, the news has picked it up a little bit as a smallpox-related virus lurking in South Texas rodents. So there's a little bit of buzz. Likely, it's probably not, we don't know, it's probably not zoonotic, but you never know. A lot of new pox viruses are being discovered that are associated with human disease. So moving on to a little bit about my vision for this wildlife disease program at AgriLife Uvalde. I can really see Texas A&M AgriLife and this wildlife disease program on the front lines of wildlife diseases that are emerging or existing involving wildlife, but also affecting humans and domestic animals. I can conduct this, I hope to conduct a high caliber research lab that combines my expertise with pathology and disease ecology and epidemiology to be able to recognize disease threats, then understand their pathogenesis, and explore the transmission cycle, and then identify targets for intervention. So I am excited to build on collaborations between existing A&M system members as well as private stakeholders, federal and state agencies, these ranches and foundations in Texas. South Texas is a really cool place to study disease because I think it's kind of a disease hotspot. There are interactions between wildlife, livestock, as well as captive exotic species. And the environment is conducive to vector survival. So vectors can overwinter, viruses persist. And there's also a lot of movement, potentially, or for sure, of animals and humans across the border. So that lends, it's also a higher risk to introduce novel pathogens or to impair attempts to clear pathogens. So at A&M specifically, there's lots of opportunity for collaboration between all the different system members and departments, as I mentioned. Also, this new global animal health research center that's being constructed is going to have BL2 and BL3 containment for large animals, including cervid species, I hear. So this could be a really good opportunity, and also for arthropod vectors within there. I could use this to look at certain wildlife species to determine susceptibility or transmission dynamics to know if something was to emerge in Texas, how would it behave in these wild populations. Also, we have IAD, which is involved also in foreign animal zoonotic disease. So I plan to have a few components for this potential research program, characterizing transmission cycles and reservoirs of disease, as I did with T. cruzi, both to identify risk factors and to inform parameters that can be put into mathematical models, as well as doing some surveillance for disease threats that are already known and then emerging, and then looking for exploring new disease concerns as they arise. So I think I'm well poised to recognize and respond to new disease threats. So I'd like to continue some of the T. cruzi wildlife research, looking at wildlife a little bit more at reservoir competence, so maybe examining a larger sample size of raccoons and coyotes, for example, but also looking at transmission efficiency. So we can detect PCR positive, there's DNA circulating in the blood, but is that animal actually capable of infecting vectors? So we can use xenodiagnosis studies with laboratory-raised triatomine bugs through collaboration to determine an actual measure of infectiousness. And then as well looking at the disease manifestations in wildlife. And I'm excited to apply a similar framework to other disease systems. So one of the disease systems I'm interested in is hemorrhagic disease. This is a vector-borne disease caused by an orbivirus, very well known in the deer world. It's been reported mostly in white-tailed deer, but also in some other species, caused either by epizootic hemorrhagic disease virus or blue tongue virus. And there are multiple molecular subtypes within those viruses, and some of them may be expanding in geographic range or emerging. New subtypes are being reported. In Texas, it appears to be enzootic, so we don't have big outbreaks for the most part. There's a high seroprevalence demonstrating the local deer have been exposed, but they're not developing disease. But introduced captive cervids that are introduced from other areas are at risk of developing hemorrhagic disease. And potentially there could be introduction of new exotic strains to the area. So it would be good to characterize better the interactions between the virus, the vector, and the host, and identify risk factors for introduction of new serotypes and their persistence. In general, these vectors, the Culicoides biting midges, are poorly understood as far as like what specific species are even present or capable or competent as vectors. And then molecular epidemiology is important to characterize better which virus, so when there is an outbreak, which virus was it and which subtype is it, and what's the distribution of these different viruses. Another system I'm really excited about is feral swine. So you guys, I'm sure, know all this. There's a ton of them around. They cause major problems. They're reservoirs of a whole bunch of diseases. This is a very short list of those. Many of them are of concern to humans or livestock, and they're potential reservoirs of foreign animal diseases that we do not want to enter the US. So feral swine, because they're susceptible to so many different pathogens, could be good sentinels for a disease. So they've been used as sentinels for West Nile virus. They're a wide-ranging, abundant species, and there's also lots of opportunities for opportunistic sampling. So these animals are harvested as part of Wildlife Services control programs. They're also routinely hunted, so we could get probably a big sample size quickly. And it would be good to look at their role as reservoirs for these different pathogens. And so I'd like to combine both serology, which can demonstrate exposure or antibodies, but not necessarily current infection, with a multiplex qPCR that could say, okay, yeah, the pathogen is actually in this animal right now, circulating, for diseases that are of highest importance. So right now, arboviruses are pretty hot. So Zika has been reported in the valley. Are the swine being exposed, or can we use them to identify other areas where this virus is circulating? Other foreign animal diseases, as I mentioned, and then other disease threats that are important to the stakeholders in South Texas. And finally, as I mentioned, I am excited and I would be well poised to explore new diseases as they arise. So I can do necropsies. I can also communicate with people in the field that have seen animals dying of unusual disease. And then I'll be able to respond rapidly to outbreaks and die-offs. And then follow that by investigating the transmission system, what are the susceptible hosts, and what vectors are involved. I think that my skills and the research I'm interested in lends itself to a diversity of funding opportunities through NIH or NSF for diseases that are of importance to human health, USDA maybe for livestock, the DHS DARPA for these emerging or foreign animal diseases, wildlife-specific funding, as well as some other smaller pots of funding available in Texas. And I'd like to acknowledge people in my lab, East Foundation for Access to Ranches, collaborators at DSHS, all the primate centers that we worked with, and then my funding support, my fellowship, wildlife, peeps, and entomology. And I'll be happy to take any questions. That's the Frio River in Uvalde. When we went to do that research, we took a day off.
Required.
Yeah. Hi.
For the Chagas disease, does it, I know there's a big distribution, through the Americas, and it's probably behaving a little differently here in Texas, but do you get a feel for, based on the players, right, involved in the system, if it's, where it is strongest along like an urban or rural, kind of gradient? I know there's a lot going on there, but your research seems to touch on a lot of things that could help you make that decision.
Yeah, so a difference between the vectors that we have in the US and those that are common in South America is that in South America, they're domesticated. They're domestic vectors. They live in the housing and exclusively almost, and feed on the inhabitants nightly. So we mostly have this sylvatic cycle. But then sylvatic in Texas is kind of like suburban areas can still be involved. So anywhere that you have raccoons and possums, you could certainly have these vectors and the pathogen. And there seems to be sort of the hotspot in Texas is hill country. It's kind of from like Austin down to San Antonio is probably where we get the most reports of vectors. But then all throughout South Texas is actually pretty common too, just less well inhabited. Yes?
Excuse me, could you explain your potential project on the xenodiagnosis? This is to characterize whether animals might be infectious.
Yeah.
And exactly how that works.
Yeah, so the lab I've been associated with and would likely continue to collaborate on some projects is Sarah Hamer's lab, and she and Gabe Hamer in entomology have established a laboratory colony of local species of triatomine bugs. So they're rearing them from eggs. So the pathogen's not passed transstadially, so the eggs are uninfected. So then they grow up and you can have known uninfected bugs. And I'm working on a project right now to use these non-human primates. We actually just got funding for this, to see if we can do indirect xenodiagnosis, so not actually have to put the bugs on the monkey, but can we get blood samples and rapidly transport them. So the bugs are fed through, just like mosquitoes are fed through blood hematech feeders. So can we put the blood from the monkeys into the colony quickly and count how many of those bugs become infected. So we would say we fed 10 bugs on known infected blood, what percentage of those actually become infected with the parasite. Anyone else?
A more general question for you.
Sure.
Clearly, you're board certified as a pathologist, and you showed histo slides and stuff. But a lot of what you're doing is also epidemiology.
Yeah.
How would you characterize yourself? Pathologist, epidemiologist, or mix, and where does your primary interest lie?
My primary interest is in disease ecology, so understanding the interactions between all the the animals and the host, and then if I can throw pathology into there to either better characterize disease or better characterize pathogenesis, that's a bonus. So most of my PhD work was not pathology focused, but I sort of pushed to say, hey, can we see if the coyotes develop disease like dogs do, because we expect them to, and it helped out with the Brazos-Pox thing as well. So.
That actually spurred up another question for me, because you talked about the extreme lesions that you saw in the coyotes. Are those comparable to the lesions that you see in dogs, or are they more severe? Or
they're comparable to some dogs that I've seen. They're less severe than others, like dogs that we see that come in dying of heart failure usually have more severe lesions. Although in some cases they actually don't, because there are functional abnormalities that can happen with the arrhythmias that don't necessarily manifest as pathologic lesions. But yeah, so they're very, so what I guess I didn't mention is we didn't actually identify the parasite in the histologic section in those coyotes, but that's also commonly seen in dogs. So a lot of times in chronic disease, you don't necessarily find the parasite, you just find this characteristic lymphoplasmacytic inflammation, and with the PCR positivity, it's highly suggestive that that's due to that parasite. Thank you. Yes?
I'm curious, Carolyn, in collecting the kissing bugs, you use UV light and searching. What was your success between the two methods?
Low on both. Probably the highest success, it depends on where you are too, it's very heterogeneous. I think the relative abundance of bugs. Highest success I personally had was down in South Texas doing the bat work, where we weren't even there looking for bugs, but we're just sitting around breathing, and having heat source, and bugs would approach us. So we had our lights turned off, waiting, then turned them on to check the nets again, and one time we're surrounded by these kissing bugs. So yeah, so the collection of the vectors is problematic for sure. When you actually go out to looking for them, it's difficult to get them. So that's why our lab has a big citizen science program where public people from all over Texas and other states actually mail us in bugs, and we've gotten way more bugs through those methods than for active searching.
We tried black lights and sheets and so forth, and we had no success with that. So
it depends where you are. Actually, at that bat cave in Concan, we did put up a sheet just to get the other little bugs away from our headlights, and we caught quite a few, not a lot, maybe 3 during that night, kissing bugs. And then out in Big Bend area, we put up a sheet, but we were also just kind of hanging around this house where they're known to arrive, and I think we got 12 in 2 hours. But most of the work that we did at the primate centers was unrewarding. So us actively searching for bugs at the Bastrop Center, we had, I think, a total of 25 hours times 3 people, but we were mostly together, we got 2 bugs. And one of them was just dead on the sidewalk, so we found it. And there's another one that we found that was alive in that same building. But none of them came to our lights. Yes?
Was that house in Big Bend K-Bar?
Yes. Yeah.
I stayed there about 10 years ago.
Yeah, so I think they don't let people, right now they're not letting people stay there anymore because they realize, yeah.
I definitely did a few times.
It's scary.
So, I may not be here.
Yeah. Yeah. I mean, luckily it's not transmitted through the bite, so there's a paper that estimated that that humans had to have 5,000 contacts with the bug before infection. You know, like total, like out of every 5,000 contacts, a human gets infected. So that's another reason probably why we don't have as much human disease in the US as in areas where the bugs are living in the house and, you know, feeding on people every night. But the oral transmission route is highly effective in dogs and probably wildlife. Okay. Thank you,
All
everyone.
right, any other questions? All right, thank you, Carolyn. All right, we're going to take